Today I'm going to talk a little bit about point-of-care medical diagnostics. And this is my website here for anyone who's interested to learn a little bit more about my lab. So medicine and the healthcare system have improved dramatically over the last 80 years. And we can see this by looking at the average life expectancy in the US. Over that time span, the average life expectancy has increased from 58 years to over 75 years. But if we look at the healthcare spending as a function of our age, we find that we disproportionately spend money in infancy and toward the later years of our lives. So it should not be surprising then that the total healthcare costs over time have been increasing dramatically. Okay, so shown here, it's a function of the GDP, and it's steadily increasing up to 17.3% recently, and projected to exceed that in the future. So I believe that the global aging population makes hospital-centric healthcare unrealistic in the future. And so my vision for that is that we need to move away from hospital-centric to home-centric healthcare. And there's two components of how we can do that. The first is telemedicine. So this is already happening with a lot of physicians where you can talk to your doctor over Skype or you can talk to your doctor on the computer and do a lot of medical applications for non-life-threatening or minor injuries. Okay? And even last week, the FDA approved an iPhone application for patients with degenerative eye conditions to track their eyesight over time. So clearly, this is a step in the right direction, and we're moving towards reducing medical costs with this. But the second approach is that we really need to move from a reactive-based medicine to proactive. So we need to be able to monitor our biomarkers over time and control how this happens. So instead of sending our samples off to a central laboratory, we move it closer to the patient where we can do point-of-care diagnostics or point-of-use diagnostics. Okay? So if we are really going to be monitoring these biomarkers routinely, we need a test that's going to be cheap and rapid and very accurate, and most importantly, doesn't require a visit to the doctor's office to complete this. So this is my large-scale vision of what I see. So we have a point-of-care biosensor where, in this case, the user has a cell phone, and we're interrogating some biological sample, whether this is blood or urine or saliva, and we're looking for a very different biomarkers in this blood that are specific to a particular disease. So obviously, these devices have to be disposable in one-time use and very easy to use. Okay? Furthermore, they should be more sensitive than the kind of home-use tests that you can find today in simple cases like pregnancy. Okay? They also need to be rapid and low cost and extremely portable. So there are many different applications for this type of a device. You know, medical diagnostics is where I focused a lot of my talk so far, and certainly is one of the biggest application spaces. But we can envision that this isn't just for bedside or doctor's office or home, but also in resource-limited settings, like developing countries where there is very little medical infrastructure already in place. So you can take this into a remote village in Africa to do disease diagnosis on site. Okay? It also has other applications such as infectious disease. We can monitor HIV, hepatitis C, tuberculosis, or even do rapid flu differentiation between H1N1 and H5N1. Environmental monitoring, you can look at pollution, groundwater, and a major one is food safety. So we can look at some of the contaminants that got into the food supply and determine that kind of at the point of sight. Another large one that seems relevant recently is biowarfare. So DARPA has done many studies that if a dirty bomb were to go off in a major metropolitan area, such as San Francisco, that in fact very few people would actually be exposed to a life-threatening exposure of radiation. However, there would be hundreds of thousands, if not millions, that were panicked from this dirty bomb. And so how do you rapidly differentiate between the people that were exposed to this potentially life-threatening radiation and those that were just panicked? And so this is one application where if you had a point-of-care device, you could rapidly test many people. And my lab's approach is that we take CMOS electronics with electronic detection and we combine it with nanotechnology and MEMS in order to develop new novel biosensing platforms. So I'm going to spend the next few minutes and talk a little bit about some of the research that I did at Stanford, and then I'll progress to what we're starting here at UCSD. So while I was a graduate student at Stanford, one of the projects we looked at was we took the magnetic sensors that were typically found in hard disk drives and we repurposed them as biosensors. So in this case, we have an array of these sensors, and on top of each sensor, we functionalized it with a capture antibody. Now this antibody is highly specific and particular to a particular biomarker. So if we're looking at HIV, this capture antibody only captures the biomarkers that are relevant in the HIV virus. Okay? We then label these with magnetic nanotags. Now the magnetic nanotags are read out by the underlying magnetoresistive sensors, which correspond to a resistance change over time if we monitor this. Now, of course, we're not just doing this on one sensor. We're doing this on an array of sensors so we can look at many different biomarkers in parallel. Okay? Oftentimes, it's not enough to just say that you've captured this one biomarker, but you need 4 or 5 to give you a true diagnosis. This is what an optical image of one of the sensors looks like. So it's actually quite a bit larger than what you would find in most hard disk drives, but it is the same materials and the same kind of underlying technology. And this is a scanning electron microscope, or SEM image, of the sensor after we've done the assay with all of the magnetic particles on it. So this is what one of the stations that I designed at Stanford looks like. And everything that I've talked about up until now is point of care. And this is kind of the definition of something that is not portable. Okay, so this lab is $20,000 to $100,000 worth of equipment, very dedicated instrumentation, and custom designed electronics. Okay? But it is not portable by any means. So one of the projects that I did was we took all of this lab's functionality and we miniaturized it into a device that's the size of a handheld paperback book. So this device allows you to do ultra-portable handheld diagnostic platform with 8-plex protein detection anywhere in the world. Okay? Overall, the weight was reduced by over 250x, and the power was reduced by over 150x. Furthermore, we got all of the integration of this device. So we have an electromagnet, we have our sensor, this is our one-time disposable use device, and all of the processing as well as the readout is contained in this self-contained device. So we demonstrated the performance of this device using P24. P24 biomarker is an inner capsid protein on the HIV virus. So we spike this protein in various different concentrations onto the device, and we see that we get readout, we get a signal proportional to the concentration of this particular biomarker. We compile all of this data into a calibration curve, and rather than display a very quantitative number to the user, that is, the exact concentration that we measured, we quartile this and display a colored light that corresponds to a particular therapy regimen. So if they're undetectable or low, this corresponds to a particular dosage of a drug that the patient should take. So in this way, we're removing any interpretation of the results and just presenting a very easy-to-use system, much like a pregnancy test. So I also mentioned at the start of my talk that we want these to be more sensitive than kind of the traditional tests. So the technology here was about 50 to 100 times more sensitive than the traditional lateral flow assays that you would find for this test. So this is some of the work that we did at Stanford, and now one of the things that my group is looking at here at UCSD is to continue this for wireless peptide arrays. So we're making a very high density sensor array, and we're performing all of our reaction on top of this for the biochemistry, and we're creating the recognition molecules here are in situ synthesized peptide arrays. Okay? And we're integrating this, or hope to integrate this, with a cell phone. So the device will be wirelessly powered and wirelessly communicate all of the data back to your smartphone, where you can do the further data analysis and compile all of the results. So I'd just like to leave with one comparison that I think is very relevant. Just as miniaturization of mainframes into the microprocessor revolutionized the computer industry, I believe that miniaturization of medical diagnostic tools will similarly revolutionize the healthcare industry. So, thank you for your time.
Do you think it's feasible to be able to detect many different things with your smartphone or with any with your device, or what's the limitation in terms of what you can and cannot detect?
Sure. So I think your question is going to, are you always going to do shotgun analysis, meaning that we don't know what we're specifically looking for, and we're just going to blindly search? And I think the answer to that is no. I think that we're always going to have very targeted diseases that we're looking for. Whether it's biomarkers that we want to monitor over time, or we're looking for specific disease sets such as HIV, hepatitis C. So we certainly can put together a panel, but I don't think it's going to be blind shotgun style.
Can you comment a little bit on the stability of the analytes that you're measuring, or the difficulty in gathering those versus the difficulty of analyzing, which seems to be a lot simplified with your research? Yeah,
so you're absolutely right. One of the main problems that you run into in some of this research is the stability of the analytes. So one of the things that we're looking at is going to peptides, which are much more stable. They have a longer shelf life than, say, antibodies. Although you could lyophilize antibodies and have them be potentially very stable over time, too.
But that's not reasonable in a developing country context, lyophilization of...
You could prepare the sticks, you could prepare the device ahead of time and ship it off as an HIV test.
Got it.
And then you're set.
Perfect. Well, thank you very much. Are there any other questions? All right, let's thank our speaker once again.